with the subject of glycogenolysis on molecular interactions relevant to cell biology at section J3, glycogen metabolism. Welcome back to the lecture. So glycogenolysis, the degradation of stored glycogen in liver and muscle cells or muscles constitutes glycogenolysis. The pathway for synthesis and degradation of oxygen are not reversible. An independent set of enzymes present in the cytosol carry out glycogenolysis. Glycogen is degraded by breaking alpha-1,4 and alpha-1,6 glycosidic bonds. The action of glycogen phosphorylase, the alpha-1,4 glycosidic bonds from the non-reducing ends, are cleaved consequentially by the enzyme glycogen phosphorylase to yield glucose 1,6-phosphate. And this process, called phosphorylysis, continues until 4 glucose residues remain on either side of branching point alpha-1,6 glycosidic link. The glycogen so formed is known as limidextrin, which cannot be further degraded by phosphorylase. Glycogen phosphorylase possesses a molecular paradoxal phosphate covalently bound to the enzyme. Okay, the action of glycogen phosphorylase. The alpha-1,4 glycosidic bonds from the non-reducing ends are cleaved sequentially by the enzyme glycogen phosphorylase to yield glucose 1,6-phosphate, 1-phosphate. This process, called phosphorylysis, continues until 4 glucose residues remain on either side of branching point alpha-1,6 glycosidic link. The glycogen so formed is known as limidextrin, which cannot be further degraded by phosphorylase. Glycogen phosphorylase possesses a molecular paradoxal phosphate covalently bound to the enzyme. Action of debranching enzyme. The branches of glycogen are cleaved by 2 enzyme activities present on a single polypeptide called a debranching enzyme, hence it is a bifunctional enzyme. Glycosyl 4:4 transferase oligo alpha-1,4 to 1,4 glucan transferase activity removes a fragment of 3 or 4 glucose residues attached at a branch and transfers them to another chain. Here, alpha-1,4 bond is cleaved and the same A1,4 bond is made, but the places are different. Amylo alpha-1,6 glucosidase breaks the alpha-1,6 bond at the branch with a single glucose residue and releases a free glucose. The remaining molecule of glycogen is again available for action of phosphorylase and debranching enzyme to repeat the reaction stated in 1 and 2. Formation of glucose 6-phosphate and glucose through the combined action of glycogen phosphorylase and debranching enzyme glucose 1-phosphate and free glucose in ratio of 8:1 are produced. Glucose 1-phosphate is converted to glucose 6-phosphate by the enzyme phosphoglucomutase. The fate of glucose 6-phosphate depends on the tissue. The liver, kidney, and intestine contain the enzyme glucose 6-phosphatase that cleaves glucose 6-phosphate to glucose. This enzyme is absent in muscle and brain, and hence free glucose cannot be produced from glucose to 6-phosphate to these tissues. Therefore, liver is the major glycogen storage organ to provide glucose into circulation to be utilized by various tissues. In the peripheral tissues, glucose 6-phosphate produced by glycogenolysis will be used for glycolysis. It may be noted that though glucose 6-phosphate is absent in muscles, some amount of free glucose, 8 to 10% of glycogen, is produced in glycogenolysis due to the action of debranching enzyme alpha-1,6 glucosidase activity. Degradation of glycogen by lysosomal acid maltase. Acid maltase or alpha-1,4 glucosidase is a lysozymal enzyme. This enzyme continuously degrades a small quantity of glycogen. The significance of this pathway is not very clear. However, it has been observed that the deficiency of lysosomal enzyme alpha-1,4-glucosidase results in glycogen accumulation, causing a serious glycogen storage disease. 2. Example: Pompey's disease. Regulation of glucogenesis, glycogenesis, and glycogenolysis. The good coordination and regulation of glycogen synthesis and its degradation is essential to maintain the blood glucose levels. Glycogenesis and glycogenolysis are respectively controlled by the enzymes glycogen synthase and glycogen phosphorylase. Regulation of these enzymes is accomplished by 3 mechanisms. Allostric regulation of glycogen metabolism. There are some metabolites that allostrically regulate the activities of glycogen synthase and glycogen phosphorylase. The control is carried out in such a way that glycogen synthesis is increased when substrate availability and energy levels are high. On the other hand, glycogen breakdown is enhanced when glucose concentration and energy levels are low. The allostric regulation of glycogen metabolism is shown in the figure. In a well-fed state, the availability of glucose 6-phosphate is high, which allostrically activates glycogen synthase for most glycogen synthesis. On the other hand, glucose 6-phosphate and ATP allostrically inhibit glycogen phosphorylase. Free glucose in liver also acts as an allostric inhibitor of glycogen phosphorylase. 2. Hormonal regulation of glycogen metabolism. The hormones, through a complex series of reactions, brings about covalent modification, namely phosphorylation and dephosphorylation of enzyme proteins, which ultimately control glycogen synthesis or its degradation. CAMP as second messenger for hormones. The hormones like epinephrine and norepinephrine and glucagon in liver activate adenylate cyclase to increase the production of CAMP. The enzyme phosphodiesterase breaks down CAMP. The hormone insulin increases the phosphodiesterase activity in liver and lowers the CAMP levels. Regulation of glycogen synthesis by CAMP. The glycogenesis is regulated by glycogen synthase. This enzyme exists in 2 forms: glycogen synthase A, which is not phosphorylated and most active, and A, which is not phosphorylated and most active, and secondly glycogen synthase B as phosphorylated inactive form. Glycogen synthase A can be converted to B form inactive by phosphorylation. The degree of phosphorylation is proportional to the inactive state of enzyme. The process of phosphorylation is catalyzed by a CMP dependent protein kinase. The protein kinase phosphorylates and inactivates glycogen synthase by converting A form to B form. The glycogen synthase B can be converted back to the degree of phosphorylation is proportional to the inactive state of enzyme. The process of phosphorylation is catalyzed by a CMP-dependent protein kinase. The protein kinase phosphorylates and inactivates glycogen synthase by converting A to B form. The glycogen synthase B can be converted back to synthase A by protein phosphatase. In the figure, the inhibition of glycogen synthesis brought about by epinephrine, also norepinephrine, and glucagon, through CAMP by converting active glycogen synthase A to inactive synthase B. Regulation of glycogen degradation by CAMP. The hormones like epinephrine and glucagon bring about glycogenolysis by the action on glycogen. Phosphorylase through CAMP. Glycogen phosphorylase exists in 2 forms: an active A form and inactive form B. The CAMP formed during hormonal stimulus activates CAMP-dependent protein kinase. This active protein kinase phosphorylates inactive form of glycogen phosphorylase kinase to active form. The enzyme protein phosphatase removes phosphate and inactivates phosphorylase kinase. The active phosphorylase kinase phosphorylates inactive glycogen phosphorylase B to active glycogen phosphorylase A, which degrades glycogen. The enzyme protein phosphatase phosphatase I can dephosphorylate and convert active glycogen phosphorylase to A to inactive B form. Effective Ca2 ions on glycogenolysis. When the muscle contracts, Ca2 plus ions are released to form from the sarcoplasmic reticulum. Ca2 plus binds to calmodulin, calcium modulating protein, and directly activates phosphorylase kinase without the involvement of CAMP-dependent protein kinase. The overall effect of hormones in glycogen metabolism is that an elevated glucagon and epinephrine level increases glycogen degradation, whereas elevated insulin results in increased glycogen synthesis. The next section is J4, hexose monophosphate shunt, interconversion of hexoses and pentoses. Hexose monophosphate pathway of HMP shunt is also called pentose phosphate pathway or phosphogluconate pathway. This is an alternative pathway to glycolysis and TCA cycle for the oxidation of glucose. However, HMP, hexose monophosphate shunt, is more anabolic in nature since it is concerned with the biosynthesis of NADPH and pentoses. HMP shunt, a unique multifunctional pathway. The pathway starts with the glucose 6-phosphate. As such, no ATP is directly utilized or produced in HMP pathway. It is a unique multifunctional pathway since there are several interconvertible substances produced which may proceed in different directions in the metabolic reactions. Location of pathway. The enzymes of HMP shunts are located in the cytosol. The tissues such as liver, adipose tissue, adrenal gland, erythrocytes, testes, and lactating mammary gland are highly active to HMP shunt. Most of these tissues are involved in the biosynthesis of fatty acids and steroids, which are dependent on the supply of NADPH. The sequence of reactions of HMP shunt is divided into two phases, oxidative and non-oxidative. Oxidative phase, glucose 6-phosphate dehydrogenase G6PD is an NADP-dependent enzyme that converts glucose 6-phosphate to 6-phosphogluconolactone. The latter is then hydrolyzed by the gluconolactone hydrolase to 6-phosphogluconate. The next reaction involved in the synthesis of NADPH is catalyzed by 6-phosphogluconate dehydrogenase to produce 3-keto-6-phosphogluconate, which then undergoes decarboxylation to give ribulose 5-phosphate. G6PD regulates HMP shunt. The first reaction catalyzed by G6PD is most regulatory in HMP shunt. This enzyme catalyzes an irreversible reaction NADPH, competitively inhibits G6PD. It is a ratio of NADPH by NAD plus that ultimately determines the flux of this cycle. Non-oxidative phase. The non-oxidative reactions are concerned with the interconversion of 3 or 4, 5, and 7 carbon monosaccharides. Ribulose 5-phosphate is acted upon by an epimerase to produce xylulose 5-phosphate, which while ribose 5-phosphate ketoisomerase converts ribulose 5-phosphate to ribose 5-phosphate. The enzyme transketolase catalyzes the transfer of 2-carbon moiety from xylulose (5-phosphate) to ribose 5-phosphate to give a 3-carbon glyceraldehyde 3-phosphate and a 7-carbon pseudoheptulose. 7-phosphate transketolase depend on the coenzyme thiamine pyrophosphate (TPP) and magnesium 2 ions. Transhydrolase brings about the transfer of a 3-carbon fragment active dihydroxyacetone from pseudohepto loose 7-phosphate to glyceraldehyde 3-phosphate to give fructose 6-phosphate and 4-carbon erythrose 4-phosphate. Transketolase acts on cellulose 5-phosphate and transfers a 2-carbon fragment to glyceraldehyde from it to erythrose 4-phosphate to generate fructose 6-phosphate and glyceraldehyde 3-phosphate. Fructose 6-phosphate and glyceraldehyde 3-phosphate can be further catalyzed to glycolysis and a citric acid cycle. Glucose may be synthesized from these two compounds. An overview of HMP shunt is given in the figure. For the complete oxidation of glucose 6-phosphate to 6CO2, we have to start with 6 molecules of glucose 6-phosphate. Of these 6, 5 moles are regenerated with the production of 12 NADPH. The overall reaction may be represented as 6 glucose 6-phosphate plus 12 NADP plus 6 water plus gives 6 CO2 plus 12 NADPH plus 12H plus 5 glucose 6-phosphate. Significance of HMP shunt. HMP is unique in generating two important products, pentoses and NADPH, needed for the biosynthetic reactions and other functions. Importance of pentoses. In the HMP shunt, hexoses are converted into pentoses, the most important thing being ribose 5-phosphate. This pentose or its derivatives are useful for synthesis of nucleic acids, RNA and DNA, and many nucleotides such as ATP, NAD+, F80, and CoA. Skeletal muscle is capable of synthesizing pentoses, although only the first few enzymes of HMP shunt are active. It therefore appears that the complete pathway of HMP shunt may not be required for the synthesis of pentoses. Importance of NADPH. NADPH is required for the reductive biosynthesis of fatty acids and steroids. Hence, HMP shunt is more active in the tissues concerned with lipogenesis, example adipose tissue, liver, etc. NADPH is used in the synthesis of certain amino acids involving enzyme glutamate dehydrogenase. There is a continuous production of H2O2 in the living cells, which can chemically damage unsaturated lipids, proteins, and DNA. This is, however, prevented to a large extent through antioxidant reactions involving NADPH. Glutathione-mediated reduction of H2O2 is given hereunder, where H2O2, the presence of glutathione peroxidase, gives water, is reduced by 2-GSH to glutathione peroxide to give GSHG oxidized, then glutathione reductase gives the production of NAD+. Glutathione reduced GSH detoxifies hydrogen peroxide. Peroxidase catalyzes the reaction. NADPH is responsible for the regeneration of reduced glutathione from oxidized one. Microsomal cytochrome P450 system in liver brings about the detoxification of drugs and foreign compounds by hydroxylation reactions involving NADPH. Phagocytosis is the engulfment of foreign bodies, of foreign particles, including microorganisms, carried out by white blood cells. The process requires the supply of NADPH. Special function of NADPH. NADPH produced in erythrocytes has a special function. And to perform, it maintains the concentrations of reduced glutathione. Reaction explained in 3, which is essentially required to preserve the integrity of RBC membrane. NADPH is also necessary to keep the ferrous ion, Fe2, plus of hemoglobin in the reduced state, so the accumulation of methemoglobin, Fe3+, is prevented. Glucose-6-pyruvate hydrogenase deficiency. G6PD deficiency is an inherited sex-linked trait. Although the deficiency in all the cells of the affected individuals, it is more severe in RBC. HMP shunt is the only means of providing NADPH in the erythrocytes. Decreased activity of G6PD impairs the synthesis of NADPH in RBC. This results in the accumulation of methemoglobin and peroxides in erythrocytes, leading to hemolysis. Clinical manifestation G6PD deficiency. Most of the patients with G6PD deficiency do not usually exhibit clinical symptoms. Some of them, however, develop hemolytic anemia if they are administered oxidant drugs or exposed to a severe infection. The drugs such as primaquine, antimalarial, acetanilide, antipyretic, sulfamethoxazole antibiotic, or ingestion of favipisin produce hemolytic jaundice in these patients. Severe infection results in generation of free radicals in macrophages, which could enter RBC and cause hemolysis. G6PD deficiency and resistance to malaria. It is interesting to know that G6PD deficiency is associated with the resistance to malaria caused by Plasmodium falciparum. This is explained from the fact that parasites that cause malaria are dependent on HMP shunt and reduce glutathione for the optimum growth in RBC. Therefore, G6PD deficiency, which is seen frequently in Africans, protects them from malaria, a common disease in this region. It is regarded as an adaptability of the people living in malaria-infected regions of the world. Well, let's just read that again. This is explained from the fact that parasites that cause malaria depend on HMP shunt and reduce glutathione for the optimum growth in RBC. Therefore, G6PD deficiency, which is seen frequently in Africans, protects them from malaria. Right, okay. So this deficiency doesn't give HMP shunt, and there's no reduced glutathione, so they don't get malaria. So glyoxylate cycle. The animals, including man, cannot carry out the net synthesis of carbohydrate from fat. However, the plants and many microorganisms are equipped with the metabolic machinery, namely the glyoxylate cycle, to convert fat into carbohydrates. The pathway is very significant in the germinating seeds, which stored triacylglycerol fat is converted to sugars to meet the energy needs. Location of the cycle. The glyoxylate cycle occurs in glyoxysomes, specialized cellular organelles, where fatty acid oxidation is also operative. Reactions of the cycle, the glyoxylate cycle, into the figure is regarded as an anabolic variant of citric acid cycle. Acetyl-CoA produced from fatty acid oxidation condenses with oxaloacetate to give citrate, which is then converted to isocitrate. At this stage, isocitrate bypasses the citric acid cycle and is cleaved by isocitrate lyase to succinate and glyoxylate. Another molecule of acetyl-CoA is now utilized to combine with the glyoxylate to form malate. This reaction is catalyzed by malate synthase, and the malate so formed enters citric acid cycle. Glyoxylate cycle is a cyclic pathway that results in the conversion of 2 carbon fragments of acetyl-CoA to 4 carbon compound succinate. The succinate is converted to oxaloacetate and then the glucose involving the reactions of gluconeogenesis. Next, we have section J5, metabolism of lipids. Lipids are indispensable for cell structure and function. Due to their hydrophobic and nonpolar nature, lipids differ from the rest of the body compounds and are unique in their action because they're hydrophobic. Triacylglycerol is the body fuel reserve. Humans, lipid constitutes about 15-20% of body weight in humans. Triacylglycerols, formerly triacylglycerides, are the more abundant lipids, comprising 85-90% of body lipids. Most of the triacylglycerols, TG, also called neutral fat or depot fat, are stored in the adipose tissue and serve as energy reserves of the body. This is in contrast to the carbohydrates and protein, which cannot be stored to a significant extent for energy purposes. Fat also acts as an insulating material for maintaining the body temperature of animals. Why should fat be the reserve fuel of the body? Why should fat be the fuel reserve of the body? Triacylglycerols are the most predominant storage form of energy. There are 2 main reasons for fat being the fuel reserve of the body. Triacylglycerols, TG, are a highly concentrated form of energy, yielding 9 cal per G, in contrast to body hydrates and proteins, and produce only 4 cal per gram. This is because fatty acids found in TG are in the reduced form. 2. The triacylglycerols are nonpolar and hydrophobic in nature, hence stored in pure form without any association with water in inert form. On the other hand, glycogen and proteins are polar. 1 gram of glycogen combines with 2 gram of water for storage. For the two reasons stated above, 1 gram of fat stored in the body yields nearly 6 times as much energy as 1 gram of hydrated glycogen. In a healthy individual weighing 70 kilograms, about 10 to 11 kilograms of fat is stored mostly in adipose tissue, which corresponds to a pure reserve of 100,000 calories. If this much of energy were to be stored as glycogen instead of fat, then the weight of the person would increase by at least 55 kgs. This explains why fat has been chosen as a fuel reserve during evolution. The fuel reserve in the form of fat stores will meet the energy requirements of several weeks of food deprivation. Hibernating animals provide good example for utilizing fat as fuel. Fatty acid oxidation. The fatty acids oxidation. These fatty acids in the body are mostly oxidized by beta oxidation. Beta oxidation may be defined as the oxidation of fatty acids on the beta carbon atom. This results in the sequential removal of the two carbon fragment acetyl-CoA. The nature of fatty acid oxidation was identified as early as 1904 by the Franz Knoop, a German scientist. Fed dogs' fatty acids tagged labeled at their last, that is omega, carbon with phenyl ring. The dogs excreted in urine a final product, phenylacetic acid, when fed even carbon fatty acids. For odd carbon fatty acids, benzoic acid was the excretory product. Knoop proposed that the fatty acids are degraded by oxidation of the beta carbon. Surprisingly, it took nearly 50 years to confirm Knoop's hypothesis. The enzymes involved in high energy or squabble oxidation and the reaction mechanism was elucidated only by 1950. The prominent biochemists who contributed to our knowledge of fatty acid oxidation include Albert Leininger, Louis Leboyer, Eugene Kennedy, Fyodor Linin, and David Green. Fatty acid oxidation stages and tissues. The beta oxidation of fatty acids involves 3 stages. Activation of fatty acids occurring in the cytosol, transport of fatty acids into mitochondria, beta oxidation proper in the mitochondrial matrix. Fatty acids are oxidized by most of the tissues in the body. However, brain, erythrocytes, and adrenal medulla cannot utilize fatty acids for energy requirements. Fatty acid activation. Fatty acids are activated to acyl-CoA by thiokinases or acyl-CoA synthetases. The reaction occurs in 2 steps and requires ATP, coenzyme A, and magnesium 2+. Fatty acid reacts with ATP to form acyl adenylate, which then combines with coenzyme A to produce acyl-CoA. In the activation, 2 high-energy phosphates are utilized since ATP is converted to pyrophosphate PPI. The enzyme inorganic pyrophosphate hydrolyzes PPI to phosphate PI. The immediate elimination of PPI makes this reaction totally irreversible. 3 different thiokinases to activate long chain 10 to 20 carbon, medium chain 4 to 12 carbon, and short chain less than 4 carbon fatty acids have been identified. Transport of acyl-CoA into mitochondria. The inner mitochondrial membrane is impermeable to fatty acids. A specialized carnitine carrier system operates to transport activated fatty acids from cytosol to the mitochondria. This occurs in 4 steps. A cell group of acyl-CoA is transferred to carnitine beta-hydroxy alpha trimethyl aminobutyrate, catalyzed by the carnitine acyl transferase 1, present on the outer surface of inner mitochondrial membrane. The acyl carnitine is transported across the membrane to mitochondrial matrix by a specific carrier protein. Carnitine acyl transferase 2, found on the inner surface of inner mitochondrial membrane, converts acyl carnitine to acyl-CoA. The carnitine released returns to cytosol for reuse. It should be noted that the coenzyme A used for activation is different from the one that finally combines with fatty acid in the mitochondria to form acyl-CoA. Thus, the cell has 2 separate pools, cytosolic and mitochondrial, of coenzyme A, inhibitor of carnitine shuttle. The carnitine acyl transferase 1 is inhibited by malonyl-CoA a key metabolite involved in fatty acid synthesis that occurs in cytosol. Details are given below. In other words, while the fatty acid synthesis is in progress, reflected by high concentration malonyl-CoA, the oxidation do not occur. Such carnitine shuttle is impaired. Beta oxidation proper. Each cycle of beta oxidation liberating a 2-carbon unit acetyl-CoA occurs in a sequence of 4 reactions. Oxidation, acetyl-CoA undergoes dehydrogenation by an FAD-dependent flavoenzyme, acetyl-CoA dehydrogenase. A double bond is formed between alpha and beta carbons, that is 2 and 3 carbons. Hydration, enoyl-CoA hydratase brings about the hydration of the double bond to form beta-hydroxyacyl-CoA. Oxidation, beta-hydroxyacyl-CoA dehydrogenase catalyzes the second oxidation and generates NADH. The product formed is beta-ketoacyl-CoA. Cleavage. The final reaction, beta oxidation, is the liberation of a 2-carbon fragment, acetyl-CoA, from acyl-CoA. This occurs by a thiolytic cleavage catalyzed by beta-ketoacyl-CoA thiolase or simply thiolase. The new acyl-CoA containing 2 carbons less than the original re-enters the beta oxidation cycle. The process continues till the fatty acid is completely oxidized. The overall reaction for each cycle of beta oxidation is CN acyl-CoA plus FAD plus NAD plus H2O plus CoASH gives CN-2 acyl-CoA plus acetyl-CoA plus FADH2 plus NADH plus H+. The scheme of fatty acid oxidation discussed above corresponds to saturated no double bond and even carbon fatty acids. This occurs most predominantly in biological systems. Oxidation of palmitoyl-CoA. The summary of beta oxidation of palmitoyl-CoA is shown here. Palmitoyl-CoA plus 7CO CoAsh plus 7 FAD plus 7 NAD plus 7 H2O gives 8 acetyl-CoA plus 7 FADH2 plus 7 NADH plus 7H. Palmitoyl-CoA undergoes 7 cycles of beta oxidation to yield 8 acetyl-CoA. Acetyl-CoA can enter citric acid cycle and get completely oxidized to carbon dioxide and water. Energetics of beta oxidation. The ultimate aim of fatty acid oxidation is to generate the energy obtained from the complete oxidation of palmitic acid, 16 carbon, is given in the table below. The standard free energy of palmitate is 22340 cals. Energy yield by its oxidation is minus 129 ATP, or is 129 to 7.3, 940 cals. Efficiency of energy conservation for fatty acid oxidation is 940 by 22340 in 140%. Okay, oxidation of odd carbon chain fatty acids. The beta oxidation of saturated fatty acids contains odd number of carbon atoms, proceeds in the same manner as described above for even carbon fatty acids. The only difference is that in the last and final beta oxidation cycle, a 3-carbon fragment is left behind in place of 2-carbon unit for saturated fatty acids. This compound is propionyl-CoA, which is converted to succinyl-CoA as follows. Propionyl-CoA is carboxylated in the presence of ATP, CO2, and vitamin birin to D-methylmalonyl-CoA. Methylmalonyl-CoA epimerase converts the methylmalonyl-CoA to L form. This reaction, dextro to levo, is essential for the entry of this compound into the metabolic reactions of the body. The next enzyme, methylmalonyl-CoA mutase, is dependent on vitamin 12 deoxyadenosyl-cobalamin. It catalyzes the conversion of methylmalonyl-CoA, a branch compound, to succinyl-CoA, a straight chain compound which can enter citric acid cycle. And now to the next part. Due to the presence of double bonds, the unsaturated fatty acids are not reduced to the same extent as saturated fatty acids. Therefore, oxidation of unsaturated fatty acids in general provides less energy than the saturated fatty acids. Most of the reactions involved in the oxidation of unsaturated fatty acids of unsaturated fatty acids are the same as those found in beta oxidation of saturated fatty acids. However, the presence of double bonds poses problems for beta oxidation to proceed. This is overcome by two additional enzymes, an isomerase and an epimerase. Beta oxidation of fatty acids in peroxisomes. Peroxisomes are organelles present in the most eukaryotic cells. The beta oxidation occurs in a modified form in peroxisomes. Acetyl-CoA dehydrogenase, a enzyme, leads to the formation of FADH2 as in beta oxidation. The reducing equivalents from FADH2 are not transferred to the electron transport chain but handed over to O2. This results in the formation of H2O2, which is cleaved by a catalase. EFADH2 plus O2 gives EFAD plus H2O, where hydrogen peroxide presents a catalase, water, and have an oxygen. There is no ATP synthesized in peroxisomal beta oxidation of fatty acids, since the reducing equivalents do not pass through He. However, heat is liberated. It is not believed that the peroxisomes carry out the initial oxidation of long-chain C20, C22, etc. fatty acids, which is followed by mitochondrial oxidation. Peroxisomal oxidation is induced by high-fat diet and administration of hyperlipidemic drugs, example cloffibrate. Zellweger syndrome. This is a rare disorder characterized by the absence of peroxisome in almost all tissues. As a result, the long-chain fatty acids C26 to C38 are not oxidized. They accumulate in tissues, particularly in brain, liver, and kidney. Hence, the disorder is also known as cerebrohepatorenal syndrome. Alpha oxidation of fatty acids. Beta oxidation is the most predominant pathway for fatty acid degradation. However, the removal of one carbon unit at a time by the oxidation of alpha carbon atom of fatty acid is known as alpha oxidation. Does not involve the binding of fatty acid to coenzyme A, and no energy is produced. Alright, and is that theta or zeta? Looks like zeta or something like that. Not able to decipher that. Okay, I think it is theta oxidation fatty acids. There seems to be a discrepancy there. Let me just check. Right, I made a reference and I understand that it is omega oxidation of fatty acids. Right, I just wanted to verify. This is a minor pathway. It involves hydroxylation followed by oxidation of omega carbon present, as a methyl group at the other end, or at one end carboxyl group is present of fatty acid. This reaction requires cytochrome P450, NADPH, and O2 besides the enzymes. Biosynthesis of fatty acids. The dietary carbohydrates and amino acids, when consumed in excess, can be converted to fatty acid and stored as triacylglycerols. De novo, new synthesis of fatty acid occurs predominantly in liver, kidney, adipose tissue, and lactating mammary glands. The enzyme machinery for fatty acid production is located in the cytosomal fraction of the acyl. Acetyl-CoA is the source of carbon atoms, while NADPH provides the reducing equivalents and ATP supplies energy for fatty acid formation. The fatty acid synthesis may be learned at three stages: production of acetyl-CoA and NADPH, conversion of acetyl-CoA to malonyl-CoA, reactions of fatty acid synthase to synthase complex. Production of acetyl-CoA and NADPH. Acetyl-CoA and NADPH are the prerequisites for fatty acid synthesis. Acetyl-CoA is produced in the mitochondria by the oxidation of pyruvate and fatty acids, degradation of carbon skeleton of amino acids, and from ketone bodies. Mitochondria, however, are not permeable to acetyl-CoA. An alternate or bypass arrangement is made for the transfer of acetyl-CoA to cytosol. Acetyl-CoA condenses with the oxaloacetate in mitochondria to form citrate. Citrate is freely transported. Acetyl-CoA condenses with oxaloacetate in mitochondria to form citrate. Citrate is freely transported to cytosol, where it is cleaved by citrate lyase to liberate acetyl-CoA and oxaloacetate. Oxaloacetate in the cytosol is converted to malate. Malic enzyme converts malate, pyruvate, NADPH, and CO to a generator in this reaction. Both of them are utilized for fatty acid synthesis. Advantages of coupled transport of acetyl-CoA and NADPH. The transport of acetyl-CoA from mitochondria to cytosol is coupled with the cytosomal production of NADPH and carbon dioxide, which is highly advantageous to the cell for the optimum synthesis of fatty acids. For the synthesis of palmitate, 8 acetyl-CoA are transported from the mitochondria to cytosol, which is linked with the synthesis of 8 NADPH in the cytosol. As such, 14 NADPH are needed for the production of 1 molecule of palmitate. The remaining 6 NADPH therefore must be obtained from HMP shunt. This sort of calculation may be more hypothetical than real, since many metabolic reactions simultaneously operate in the cell. Now, the formation of malonyl-CoA. Acetyl-CoA is carboxylated to malonyl-CoA by the enzyme acetyl-CoA carboxylase. This is an ATP-dependent reaction and requires biotin for carbon dioxide. And fixation. This is an ATP-dependent reaction and requires biotin for carbon dioxide fixation. The mechanism of action of acetyl-CoA carboxylase is similar to that of pyruvate carboxylase. Acetyl-CoA carboxylase is a regulatory enzyme, in fatty acid synthesis. Reactions of fatty acid synthase complex. The remaining reactions of fatty acid synthesis is catalyzed by a multifunctional enzymes known as fatty acid synthase (FAS) complex. In eukaryotic cells, including man, the fatty acid synthase exists as a dimer with 2 identical units. Each monomer possesses the activities of 7 different enzymes and an acyl carrier protein ACP bound to 4'-phosphopentathene. Fatty acid synthase functions as a single unit catalyzing all the 7 reactions. Dissociation of the synthase complex results in loss of the enzyme activities in the lower organisms, prokaryotes. The fatty acid synthesis is carried out by a multi-enzyme complex in association with a separate acyl carrier protein. This is in contrast to eukaryotes. Where ACP is a part of fatty acid synthase. The sequence of reactions for extramitochondrial synthesis of fatty acids, palmitate, is depicted in the figure and described below. The 2-carbon fragment of acetyl-CoA is transferred to ACP of fatty acid synthase, catalyzed by the enzyme acetyl-CoA ACP transacylase. The acetyl unit is then transferred from ACP to cysteine residue of the enzyme. Thus, ACP site falls vacant. The enzyme malonyl-CoA ACP transacylase transfers malonate from malonyl-CA to bind to ACP. The acetyl unit attached to cysteine is transferred to malonyl group. Bound to ACP, the malonyl moiety loses CO2, which was added by acetyl-CoA carboxylase. Thus, CO2 is never incorporated into fatty acid carbon chain. The decarboxylation is accompanied by the loss of free energy, which allows the reaction to proceed forward. This reaction is catalyzed by beta-ketoacyl ACP synthase. Beta-ketoacyl ACP reductase reduces ketoacyl group to hydroxyacyl group. The reducing equivalents are supplied by NADPH. Beta-hydroxyacyl ACP undergoes dehydration. A molecule of water is eliminated and a double bond is introduced between A and 1.3 carbons. A second NADPH dependent reduction catalyzed by aryl ACP reductase occurs to produce acyl ACP. The 4-carbon unit attached to ACP is butyryl group. The carbon chain attached to ACP is transferred to cysteine residue, and the reactions 2 to 6 are repeated 6 more times. Each time the fatty acid chain is lengthened by a 2-carbon unit obtained from malonyl-CoA. At the end of 7 cycles, the fatty acid synthesis is complete and 16-carbon fully fatty acids, namely palmitate bound to ACP, is produced. The enzyme palmitoyl thioesterase separates palmitate from fatty acid synthase. This completes the synthesis of palmitate. Summary of palmitate synthesis. Of the 16 carbons present in palmitate, only 2 come from acetyl-CoA directly. The remaining 14 are from malonyl-CoA, which in turn is produced by acetyl-CoA. The overall reaction of palmitate synthesis is summarized: 8 acetyl-CoA plus 7 ATP plus 14 NADPH plus 14H gives palmitate plus 8 CoA plus 7 ADP plus 7 pi plus 6 water. Fatty acid synthase complex. The diagrammatic representation of the model for fatty acid synthase FAS multi-enzyme complex is depicted here. It's this one. Now this model is tentative and is largely based on the work of Wakio. Fatty acid synthase is a dimer composed of 2 identical subunits, monomers each with a molecular weight of 440,000. Each subunit contains the activities of 7 enzymes FAS and an ACP with 4'-phosphopantethene SH group. These 2 subunits lie in the antiparallel head-to-tail orientations. The SH group of phosphopantethene, one subunit, is in close proximity to the SH of cysteine residue of the enzyme ketoacyl synthase of the other subunit. Regulation of fatty acid synthesis. Fatty acid production is controlled by enzymes, metabolites, end products, hormones, and dietary manipulations. Some of the important regulatory mechanisms are discussed here. Acetyl-CoA carboxylase. This enzyme controls a committed step in fatty acid synthesis. Acetyl-CoA carboxylate exists as an inactive protomer, monomer, or an active polymer. Citrate promotes polymer formation. Hence, fatty acid synthase, on the other hand, palmitoyl-CoA and malonyl-CoA cause depolymerization of the enzymes, therefore inhibits fatty acid synthase. Hormonal influence. Hormones regulate acetyl-CoA carboxylase by a separate mechanism, phosphorylation in active form and the dephosphorylation active form of the enzyme. Glucagon, epinephrine, norepinephrine inactivate the enzyme by CAMP-dependent phosphorylation. Insulin, on the other hand, dephosphorylates and activates the enzyme. Thus, insulin promotes fatty acid synthesis, while glucagon inhibits. Each monomer of FAS contains all the enzyme activities of fatty acid synthesis, but only the dimer is functionally active. This is because the functional unit consists of half of each subunit interacting with the complementary half of the other. Thus, the FAS structure has both functional division and subunit division. The 2 functional subunits of FAS independently operate and synthesize 2 fatty acids simultaneously. Functional significance of FAS complex. The organization of different enzymes of a metabolic pathway into a single multi-enzyme functional unit has distinct advantages for cellular function. The FAS complex offers great efficiency that is free from interferences of other cellular reactions for the synthesis of fatty acids. Since the entire process of the metabolic pathway is confined to the complex, there are no permeability barriers for the various intermediates. The multi-enzyme polypeptide complex is coded by a single gene. Thus, there is a good coordination in the synthesis of all enzymes of the FAS complex. And the dietary. Insulin stimulates tissue uptake of glucose and conversion of pyruvate to acetyl-CoA. This also facilitates fatty acid formation. Dietary regulation. Consumption of high carbohydrate or fat-free diet increases the synthesis of acetyl-CoA, carboxylase, and fatty acid synthase, which promote fatty acid formation. On the other hand, fasting or high-fat diet decreases fatty acid productions by reducing the synthesis of these two enzymes. Availability of NADPH. The reducing equivalents for fatty acid synthesis are provided by NADPH, which come under either the citrate acetyl-CoA transport or hexose monophosphate shunt HMP. About 50 to 60% of required NADPH is obtained from HMP shunt, which influences fatty acid synthesis. Desaturation of fatty acid chains. A microsomal enzyme system called fatty acyl-CoA desaturase is responsible for the formation of unsaturated fatty acids. This reaction also involves flavin-dependent cytochrome Bs reductase, NADH, and molecule O2. The monounsaturated fatty acids, namely oleic acid and palmitoleic acid, respectively synthesized from stearate and palmitate. Mammals lack the ability to introduce double bonds in fatty acids beyond carbons 9 and 10. Hence, linoleic acid 18, 2, 9, and 12, and linoleic acid 18, 3, 9, 12, and 15 are essential for man in the diet. However, arachidonic acid 24, 5, 8, 11, 14 can be synthesized from linoleic acid by desaturation chain elongation. Arachidonic acid is the precursor for eicosanoids, prostaglandins, and thromboxanes, a group of compounds with diversified functions. Synthesis of long-chain fatty acids from palmitate. Palmitate is the end product of the reactions of fatty acid synthase system that occurs in cytosol. Further chain elongation can take place either in the mitochondria or in endoplasmic reticulum microsomes. By separate mechanisms, the microsomal chain elongation is more predominant and involves successful additions of malonyl CoA with the participation of the NADPH. These reactions are similar to that catalyzed by fatty acid synthase. A specific group of enzymes, namely elongases, bring about fatty acid chain elongation. And the mitochondrial chain elongation is almost a reversal of beta oxidation of fatty acids. Acetyl-CoA is successively added to fatty acid to lengthen the chain. The reducing equivalents are derived from NADPH. Comparison between fatty acid synthesis and oxidation. The synthesis of fatty acids and the oxidation are two distinct and independent pathways. A comparison between these two metabolic pathways is given in the table below. Show major tissues. Fatty acid synthesis would be from liver, adipose tissue, beta oxidation, muscle, and liver. Subcellular site is cytosol, beta oxidation, mitochondria. Precursor and substrate, acetyl-CoA, acyl-CoA. End product is palmitate, is acyl-CoA. Intermediates are bound to acyl carrier protein, coenzyme A. Coenzyme requirement, NADPH supplying reducing equivalents. FAD and NAD plus gets reduced. Carbon units added, degraded, malonyl-CoA, acetyl-CoA. Transport system citrate, mitochondrial cytosol, carnitine cytosol mitochondria. Inhibitor long chain acyl-CoA inhibits acetyl-CoA carboxylase. Here malonyl-CoA inhibits carnitine acyl transferase 1. The pathway increased after rich carbohydrate diet in starvation. Hormonal status that promotes is high ratio of insulin or glucagon, low ratio of insulin glucagon. Status of enzymes, multifunctional enzyme complex, and individual enzymes. So fatty acid synthesis and the beta oxidation, what you heard. Synthesis of triacylglycerols, TG. Synthesis mostly occurs in liver and adipose tissue and to a lesser extent in other tissues. Fatty acids and glycerol must be activated prior to the synthesis of triacylglycerols. Conversion of fatty acids to acyl-CoA by thiokinase is already described. Synthesis of glycerol 3-phosphate. Two mechanisms are involved for the synthesis of glycerol 3-phosphate. In the liver, glycerol is activated by glycerol kinase. This enzyme is absent in adipose tissue. In both liver and adipose tissue, glucose serves as a precursor for general glycerol 3-phosphate dihydroxyacetone phosphate DHAP, producing glycolysis reduced by glycerol 3-phosphate dehydrogenase to glycerol 3-phosphate. Addition of acyl groups to form TG. So the remaining portions after this will be continued in the next lecture. Glycerol 3-phosphate, acyl transferases catalyzes the transfer of an acyl group to produce lysophosphatidic acid. DHAP can also accept acyl group, ultimately resulting in the formation of lysophosphatidic acid. Another acyl group is added to lysophosphatidic acid to form phosphatidic acid, 1,2-diacylglycerol phosphate. The enzyme phosphatase cleaves a phosphate of phosphatidic acid to produce glyacylglycerol. Incorporation of another acyl group finally results in synthesis of triacylglycerol. The three fatty acids found in triacylglycerol are not of the same type. A saturated fatty acid is usually present on carbon 1, and unsaturated fatty acid is found on carbon 2, and carbon 3 may have either. The intermediates of TG synthesis, phosphatidic acid and diacylglycerol, are utilized for phosphatidic